Hello students, in continuation with our series on recombinant DNA technology, now I will talk about the manipulation of purified DNA and basically I will continue with it. I have talked about this manipulation of purified DNA in a previous lecture where we talked about the restriction endonucleases, that the DNA that we have isolated, the sources, the different sources of DNA, the plasmid DNA, the genomic DNA or the bacteriophage DNA that we have isolated, how we can manipulate these purified DNA and further process to become, to make the or to generate the recombinant DNA. So, the specific learning objectives would be to understand the tools for DNA manipulation. Particularly, we will talk about the characteristics of ligases, the characteristics of polymerases and the characteristics of various modifying enzymes to modify the DNA that has been cleaved by the restriction digestion. The references for this particular section would be the T.A. Brown, which is a gene cloning and DNA analysis and introduction to T.A. Brown by 6th edition, and a Twyman book, The Advanced Molecular Biology, a concise reference. The references are used for the educational purposes only. Now, I will just review that for the construction of recombinant DNA, we require to manipulate the purified DNA samples, both the vector and our gene of interest or the fragment DNA to be cloned. Now, this involves cutting, joining as well as shortening, lengthening, copying into RNA or new DNA molecules and modification by the addition or removal of specific chemical groups. Now, DNA manipulative techniques make the use of purified enzymes. DNA manipulative enzymes can be grouped into 4 broad classes depending on the type of reaction that they catalyzed. So, there are nucleases. These are the enzymes that cut, shorten or degrade the nucleic acid molecules. And as I said, for making a recombinant DNA, we need to have 2 DNA from different sources and we need to put them together. So, to put them together, we need to cut these 2 DNA molecules and then ligate them together. So, for cutting, these nucleases are used, and in another section we have already talked in detail about the restriction endonucleases which cut the DNA at specific sites. We use ligases that will join the nucleic acid molecules together, the polymerases that will make the copies of the molecules, and the modifying enzymes that will remove or add the chemical groups. So, we will talk about these 3 categories in this section. So, ligases are the molecules that will join the nucleic acid molecules together. In the cell, the function of the DNA ligase is to repair the single-stranded breaks or what is called as discontinuities that arise in double-stranded DNA molecules during, for example, DNA replication. So, there is always a spontaneous occur of such kind of breaks that can occur, the discontinuities that can occur. So, discontinuity basically means the breakage of the phosphodiester bond. So, this is there are different terminologies which are used for the break in the DNA molecules, because when in this particular book, the discontinuity is called by the breaking of the phosphodiester bond. Nick is called when there are loss of nucleotides also. So, there is a break and there are some loss of the nucleotides. Whereas, break is called or the cut is called when both the strands of the DNA are cut. So, discontinuities can be only on the one strand or can be on both the strands, but that will only break the phosphodiester bond. Nick can be on one of the strands where there is a breaking and as well as loss of a particular amount of nucleotides, 2, 3, 4, whatever. So, that gap is created and that is called as a nick. Whereas, a break or a cut will be when there will be breaking in both the strands, and it can be just a discontinuity or it can be loss of the nucleotides also. So, now they can spontaneously occur during the processes such as DNA replication. Now, ligases, the DNA ligase in the cell works to repair these nicks or the breaks or the discontinuities. Basically, the discontinuities, because the function of the ligase is to make the phosphodiester bond. They can work in collaboration with other enzymes to fill the gaps also or to rejoin the breaks also. So, ligases can join together 2 individual fragments of double-stranded DNA. So you can see on the left hand side there is a discontinuity which is there, and it can be repaired by the DNA ligase. Or if this discontinuity is basically on both the strands, then it can basically join these two molecules also. So a discontinuity is quite simply a position where a phosphodiester bond between the adjacent nucleotides is missing. Now ligation is the final step in construction of a recombinant DNA molecule. So what happens is that we have used the vector, we have digested it with a particular enzyme, we are having a clone, gene of interest that we need to clone inside this vector, and this joining is facilitated by the DNA ligase enzymes. Now we will deal with this later, that what are the ends, what are the possibilities that these two ends can be joined or not. But basically when there are two nucleotides, which one has a 5' phosphate group and the other has a 3' OH group, then the phosphodiester bond can be formed between them. That is the simple rule. So DNA ligase can facilitate that. Now if they have one at the 5' phosphate end and 3' hydroxyl end, then the DNA ligase can make a phosphodiester bond between it, whether that is a same molecule that is cut or whether they are two different molecules that are cut by different enzymes. So therefore when you are recombining two different sources, then the DNA ligase can actually ligate it as shown in the figure. So ligation of sticky end molecules. Now when we talked about restriction endonucleases, we talked that restriction endonucleases can actually produce two different types of ends. If they cut in a staggered way, where they cut, the cutting position on one strand is different from the cutting position on the second strand, then they would give rise to what are called as the sticky end molecules, which are just shown here, which will have overhangs. So it can be a 5' overhang or it can be a 3' overhang, but it will be a overhang. Overhang basically means that single stranded portion which is there. So you can see that the restriction enzyme is produced sticky ended molecules, and DNA ligase can actually join these sticky end molecules together. Now ligation of sticky ended molecules is a very efficient process, because the complementarity nature of the overhangs will actually bring it to the proximity and keep it for a longer time in that manner, so that the ligase can very efficiently work and make the phosphodiester bond to join these two molecules together. So if the vector and the gene of interest or the fragment of interest is digested in such a manner that they produce sticky end molecules, then the generation of recombinant molecule is more efficient than when there are no sticky end molecules which are there. So sticky ends increases the efficiency of ligation. And why? Because the compatible sticky ends can base pair with one another by hydrogen bonding, forming a relatively stable structure for the enzyme to work on. Now look at if there are blunt end molecules. Now blunt end molecules, they do not, that is not a very efficient ligation, because they don't have such overhangs which will, on based of the complementarity or the compatibility, they will not come to the proximity of each other. So the ligation will have to be a chance process, that the two ends will come together and therefore the ligase will make a phosphodiester bond. Now this is circumvented by using a higher concentration of the insert molecules as well as higher concentration of DNA molecules which are there. But that is why the ligase is unable to catch hold of the molecules to be ligated and has to wait for chance associations to bring the ends together. So blunt ended molecules a little difficult, but sometimes we can't help it. We have to have a blunt end ligation because we have to use certain enzymes. So it depends upon that what vector we are using, what enzyme sites are available in that vector, or what enzyme sites we want to cut our DNA fragment with. Because sometimes you cannot use the common enzymes which are used to produce sticky end molecules, because the gene of interest will also contain those sites. So if you cut the gene of interest with that particular site, it is going to cut your gene also. So it depends upon like what sites are present in your genes, or what idea do you have about it, or what idea you can make about it by cutting and running it the fragments, that what size you are getting in. So based upon that, you have to choose sometimes the restriction enzymes which will produce blunt end molecules. Now to make this efficient, one can put the sticky ends onto a blunt ended molecule, and this is done by, for example, using linkers. Now linkers are short pieces of double stranded DNA of known nucleotide sequences that are synthesized in the test tubes. So certain oligonucleotides, double stranded molecules, can be created and generated, and these can then be ligated to the blunt end molecules, so that when you cut this linker, it will give rise to the sticky ends. So for example, this particular linker is shown here. It's a double stranded molecule containing a BAM-H1 site, and it has been created in vitro. Now if you add these linkers to the DNA fragment with blunt ends, then the linkers will join together at the end, and when you cut it with the BAM-H1, you will have the sticky ends to the blunt ended DNA fragment. Now one can say that when you are ligating the linker to the blunt end fragment, then that is also a blunt end ligation, because you will use the DNA ligase to ligate it. Now it will be still an efficient process, because you can use large concentration of linker molecules, so that the probability of joining the linkers at the ends of the DNA fragment is high. So higher concentration of the DNA fragment of interest as well as the vector sometimes is not possible. So in this case, because linkers are shorter DNA fragments, then you can probably use large concentration of these linkers. So the probability of linker joining is higher, and then you can digest it with the site which is in the linker, and you can have the sticky ends which will be efficient to join into the vector or the other molecules. Now one problem with the linkers is that if that particular site which you are using, which the linker is carrying, is also present in the DNA fragment. So for that, adapters are used. So adapters are again short synthetic oligonucleotides synthesized so that it already has one stick end. So adapter will typically have an overhang only, it will be a cut site, it will have a BAM-H1 sticky end by itself. So what will do is, they will clone this particular fragment from the blunt end site onto the blunt end fragment, which needs to have the sticky end, and after this ligation you will have the sticky end in gas. But again the problem is that because the sticky ends are compatible, then the adapters can join towards themselves. One adapter can join to the other adapter molecule and give rise to such a double stranded molecule, and it will not join to the blunt end fragment where we want it to join. So to circumvent this problem, what is basically done, so it can happen in such a manner, then when you are using the adapters, then the linkage can occur between the two adapter molecules. Now it is important to note here that the phosphodiester bond is formed between the 5' phosphate and the 3' OH. So 5' phosphate and 3' OH is essential for the ligase to work. Now if we remove the 5' phosphate, for example, and make it the 5' OH group only, then the two adapters cannot join together. So that is what is done basically. So the adapter molecules are prepared. So the blunt end side will have the phosphate and the OH group, whereas the non-blunt or the sticky end side is modified so that both the 5' as well as the 3' end will have the hydroxyl groups. Now the two linkers will not join together from the sticky ends, but the blunt end will only join to the fragment. So this modification can be done by using the modifying enzymes, as we will see subsequently, and then this can be ligated, and after ligation, again the phosphate groups can be added onto the sticky end 5' side so that they can be cloned into the vector or as required. So as it is shown, at first the adapters will have hydroxyl group both on the 5' as well as the 3' side towards the sticky end. The blunt end side will join to the DNA fragment, the blunt end DNA fragment, And then we use a modifying enzyme called as polynucleotide kinase, which will add the phosphate group to the 5' side. So now we have attained our fragment with 2 sticky ends and ready to join any other DNA fragment. So this can also be done to have the sticky ends, which is an efficient process. Another way by which ligation can be done is the homopolymer tailing. A homopolymer is a polymer in which all the subunits are the same. For example, polydeoxyguanosine or poly-DG as it's written as, or polycytosine or polydeoxythymine, it can be anything, homopolymer. The tailing involves using the enzyme, which is again a particular modifying enzyme called as terminal deoxynucleotidyl transferase, to add homopolymers onto the 3' OH termini of a double-stranded DNA molecule. So homopolymer tailing is basically when there is no compatibility between the two DNA that you want to join. So in that case, what is basically done is that a continuous stretch of a same subunit DNA molecule is added onto one end. For example, like this. So you synthesize a homopolymer tail, and this homopolymer tail is then added onto the sides of the particular DNA. Now it is important to note that the two different sources of DNA will have the opposite nucleotides, the nucleotides which are compatible to each other, tails added to their ends. For example, if one of the DNA is having a poly-DG tail, then the other DNA will have a poly-DC tail. So that when these two are brought together, then the continuous G subunits will be compatible to the continuous C subunits, tails, which have been added to the other DNA, and the two strands can be joined. So this basically solves the problem if we cannot have, or if we cannot join the two fragments and the blunt end ligation is giving a very much problem. So it is better to add the homopolymer tails, make single stranded of these tails, and then compatible ends will again come together and the ligase can do its job very nicely. So this is again shown here that how it can be done. So there is a basically the tailing is there, and you can see that there is a this tail might not be of equal size. If the two tails are of not equal size, when they come together, they will give rise to a neck. And this neck can then be filled with other, the copying, the polymerase enzymes, it can be filled by that enzyme. Now, if the complementary homopolymer tails are greater than 20 nucleotides, then quite stable base paired associations are formed. A recombinant DNA molecule held together by base pairing, not completely ligated, is often stable enough to be introduced into the host cell. Once inside the host, the cell's own DNA polymerase and DNA ligase repair recombinant DNA molecule, completing the construction that is begun in the test tube. So what this basically means is that if you are adding the homopolymer tails, then after the adding of the homopolymer tails and the compatibility, you need to still use the ligase to make the phosphodiester bond. But that is if the two homopolymer tails are of equal size. If the homopolymer tails are not of equal size, then a gap will be created. There will be certain nucleotides that will be missing. That can also be taken care of by using a DNA polymerase that will fill the gap, and then using the ligase to finally ligate it together. Now, if the homopolymer tails are very much longer in size, then they will still join, they hold each other in a very tight manner that they can easily be transformed, because the next step after the formation of recombinant DNA will be to introduce it into a host. So even without using the polymerase and even without using the ligase, these molecules where the homopolymer tails are joining them together, the two molecules, holding them together, they are not yet joined, but holding them together, they can be introduced into the host, and then the host enzymes, the polymerase as well as the ligase, can fill the gap as well as ligate the DNA molecule to make a whole recombinant DNA molecule. So, this homopolymer tailing again becomes a very important method to combine the two different sources where the compatibility of the ends cannot be achieved by any other method. Now, we come to the polymerases that makes the copies of the DNA molecules. DNA polymerases are the enzymes that synthesize a new strand of DNA complementary to an existing DNA and RNA template, and we have talked about them in detail in the molecular biology when we talked about the DNA replication. Now, these polymerases, we can have a the intact DNA polymerase has a 5' 3' nuclease, nucleotide synthesis activity as well as the 3', 5' exonuclease activity. So, it will basically run over and it will degrade whatever the incident, the original strand is and synthesize a new strand. There are two different components in this enzymes that catalyze the nucleotide synthase activity as well as the exonuclease activity. So, the part which only catalyzes the nuclease synthesis activity has been purified and it is called as the clinofragment. That will only fill the gap and will not override the existing DNA fragment. So, that can also be used to copy the DNA molecules. So, an important part is to generation for the blunt end fragments is to do the end filling and the trimming by the use of these fragments. So, these are the different possibilities that can occur in the two different sources of DNA which we are using. So, for example, in the first case there are overhangs, there are 5' overhangs and these overhangs can be degraded by the 3' to 5' exonuclease activity of the polymerases and they will generate the blunt ends that then can be ligated. You have two incompatible sticky ends, which you have trimmed to make the blunt end fragments that can then be ligated. In the second case, for example, if you have 3' overhangs, then for the 3' overhangs, the clinofragment can synthesize the end filling can be done and then generate the two blunt end fragments that can then be ligated. In the third case, then the degradation can also occur. For example, the S1 nuclease can degrade the 5' overhangs also, which can then generate the blunt end fragments and which can then be ligated. So, there is a possibility that the incompatible ends which you get can be trimmed or end filled to produce the blunt end fragments and then the ligation can be proceeded. So, another enzyme which is for the synthesis is the reverse transcriptase, which uses the RNA as the template and synthesize the new DNA molecule. And this is used when we want to make the recombinant DNA containing the cDNA or the DNA which is produced by using this enzyme from the RNA transcript. So, when we need to learn the expression level, then we use this reverse transcriptase enzyme. Another enzyme used for synthesis is the Taq DNA polymerase, which is isolated from the Thermus aquaticus thermophile. This DNA polymerase is used in the polymerase chain reaction as it is thermoresistant and it can withstand higher temperatures, and PCR requires the DNA to be put into higher temperatures where the melting and the separation of the two DNA strands can occur, and there the synthesis is carried out by the Taq DNA polymerase, which is a thermophilic DNA molecule. Lastly, we come to the modifying enzymes that remove or add chemical groups. In this, alkaline phosphatase is very important. This alkaline phosphatase removes the phosphate group from the 5' so that two hydroxyl groups are put at the ends. We have already talked about these two hydroxyl groups, for example, when we didn't want the two adapters to link together. Similarly, if we do not want the vector molecule to recircularize, we can cut it by the hydroxyl. We can use the alkaline phosphatase to remove the 5' phosphate group and become hydroxyl group. So, the vector will not auto-ligate, but it will only ligate if the phosphate group comes to the OH group, and later on the ligase can do its job and we can use the next enzyme, which is the polynucleotide kinase. So, alkaline phosphatase is an important molecule. Polynucleotide kinase, which is again isolated from E. coli T4-Phage, which has the reverse effect to alkaline phosphatase, it adds the phosphate groups onto the 5' termini. So, when the ligase has to be used and when the alkaline phosphatase has been already used, then we need to reverse it by using the polynucleotide kinase. We can carry on this reaction in the tube, or we can, as I talked about, the cell's machinery can also take care of it if the two strands are stable enough to be held together and transformed, then the kinase of the cell can also do this job and the ligase of the cell can also do it. The last enzyme in this series is the terminal deoxynucleotidyl transferase. Again, we have talked about it. They transfer or adds one or more deoxyribonucleotides onto the 3' terminus of a DNA molecule. So, when basically end filling has to be done, or new molecules have to be ended at the end. So, the single stranded molecules, the polynucleotides are added, or to the double stranded molecules, the polynucleotides have been added. The nucleotides are added, the transferred nucleotides are only from the 5' to 3' direction. So, they can give rise to the desired ends that can be then ligated to form a recombinant DNA molecule. So, this completes the manipulation of the DNA, the purified DNA that we have done, and next we move on to the formation of the recombinant DNA and then transformation into the host molecules in our next session. Thank you.